Okay. All right, I'm going to go ahead and kick this off with introducing the introductions. So today, thanks for coming. We have what I would say is our emerging theme today is topics in developmental genetics, regulation of development, coming at it from two very widely different angles. And we'll begin with by Rachel.
Hello, everyone. My name is Rupa Sridharan. I have the pleasure of being Xiaoya Zhang's mentor. So, as some of you might know, my lab is very interested in how cell fate is controlled at the level of histone modifications and chromatin accessibility. So, for the past few years, I've been obsessed with this protein called DOT1L, and Xiaoya has bravely taken on a project. And one of the model systems we use in my lab to study how cell fate is determined is a process of reprogramming of somatic cells to things called induced pluripotent stem cells. And there are lots of, the whole epigenome is actually very different in pluripotent cells than somatic cells. And so what we've been doing in my lab is to try and increase the efficiency of this conversion, but by making the chromatin environment of the somatic cell more like that of a pluripotent cell. So one of the modifications that's very different, as Xiaoya will tell you, is a particular modification. I won't go into what it is, but the problem with these histone modifications is that they are written by a whole slew of enzymes, and they're also erased by a whole slew of enzymes. Unlike the one modification that Xiaoya studies, which is K79 methylation. So I was very excited to follow this up because there's only one enzyme that writes this modification, and there is no known demethylase. So I was like, great, now we get rid of this one protein and we're going to know exactly how this modification functions. And as always in science, I was wrong. But Xiaoya has made some really fun discoveries along the way. And I also have to add that it's truly a pleasure to be Xiaoya's mentor because she keeps me on my toes. Every personal meeting we have, she always challenges my hypothesis and always has an alternative. So it's wonderful to be her mentor because of her capability as a wonderful scientist. So with that, I'll let her take it away.
Thank you, Rupa, for the wonderful introduction. So today I'm very happy to share my project on the function of DOT1L in cell fate determination. The first, as we know, multicellular organism will need cells with different functions, and all those cells are coming from one single zygote, and that can divide and become a blastocyst. At this stage, you can take the, let me open the laser. You can take the inner cell mass of the blastocyst and culture in vitro, and that one can self-renew infinitely, and it can differentiate into all three different germ layers. And eventually they can become somatic cells that has distinct functions. And during this process, the cells will lose its developmental potential, but they're gaining different function and commit different lineages. So this is a very highly dynamic change which can be regulated by epigenetic modifications, including modification on the DNA and on histones. Histone modifications are known to be associated with active or repressive gene expression. So first we want to ask, is there a global difference of histone modifications in the pluripotent cells and differentiated somatic cells? So here, Rupa performed a quantitative mass spec experiments on the pluripotent stem cells, and here we use the mouse embryonic fibroblast meth. So when we compare, we can see some histone modifications that are associated with, known to be associated with active gene expression, like acylations are much more enriched in pluripotent stem cells compared to somatic cells. And in the same mass spec data, this table is showing the fold change of somatic cells compared to stem cells. So here we can see, as expected, because kind of like reversely, you can see that the repressive histone marks are much more enriched in somatic cells compared to stem cells, where they have committed to a certain lineage and closed most of their programs and only express certain functions. But surprisingly, the most different marks in the MEFs compared to stem cells are actually associated with transcriptional elongation that are found on active genes, on the gene body of active genes. And among those modifications, K79 dimethylation is the most different, which is 7-fold higher in somatic cells than stem cells. This is a very this high enrichment in the in somatic cells led us to think, does it have a function in the self-determination? Why does it change so much during cell differentiation? And just to give you a little bit of background about K79 methylation, this is an elongation-associated mark that is found on the gene body of active genes near the transcriptional start site. And from here, I'm going to use H3K79ME to refer to all degrees of K79 methylation, mono, di, and tri. So in mammals, the tri have the least abundance, and the K79 di is found have a higher percentage, and mono has the most prevalent histone modifications among the three. And it is found in most expressed genes in the genome. So it is quite universally expressed, deposited on the genes that are expressed. And back to our title, so DOL1L is the only methyltransferase of all three different levels of K79 methylation, mono, di, and tri. So it is mainly a homolog of DOL1, which is disruptor of telomeric silencing 1. It's first described in yeast, and it is a distributed enzyme, which means every time it has to come off the chromatin before it adds the next method group. So the level of methylation, mono, di, and tri, is totally dependent on like how often the frequency of DOL1L being recruited to the certain location. And it has the catalytic domain, which is adding the method group, and also have regulatory domain that interacts with different proteins and like NRS that gives the signal. And as Rupa mentioned, there's not validated demethylase for K79. It can be removed by nucleosome turnover. So a methylated histone can be removed from the chromatin during transcription or or DNA replication. And so in this model, because methylation can only be added by one single protein and no demethylase, so we can manipulate DOL1L and study the K79 methylation in the self-determination. So we have seen in the mass spec data that K79 methylation is much more enriched in somatic cells compared to stem cells. So we are asking, is there really a function of DOL1L of this modification during the self-determination? And to answer that question, we're going to look at the importance of DOL1L in 3 different models. So our lab used pluripotent stem cells, and a cell can be differentiated into somatic cells, and we also use reprogramming. So in the lab, reprogramming can serve as a great model for studying the pluripotency acquisition. So we can induce the expression of Yamanaka factors, the OSKM transcription factors, in the mouse embryonic fibroblast MEFs. And this can activate certain programs and reprogram the somatic cells back into a pluripotent state that is functionally equivalent to embryonic stem cells. So we call it induced pluripotent stem cells. And then that one can express pluripotent markers like NANOG. So we can stain with nano antibody and use the NANOG colonies as an indicator for reprogramming efficiency. So from the mass spec, we know Cas9 methylation are lower in stem cells. So what happens if we inhibit DAW1L, and does it improve reprogramming? This experiment has been done by a postdoc in the lab, Coral. She performed reprogramming with or without DAW1L inhibition, where we used a small chemical molecule. And we can see that upon the DAW1L inhibition, there's a drastic improvement of reprogramming efficiency measured by the nanopositive colonies. And so Cas9 methylation here seems to be a barrier for programming. So then, do pluripotent stem cells need Cas9 methylation? Different studies have shown that DAW1L deficient stem cells are actually viable, and they're still expressing pluripotent markers. Like here is like rough assay of the pluripotency marker on the cell surface called alkaline phosphatase. So if you knock out or catalytic or inactivate DAW1L, you see no big difference in growth rates or in the AP staining. Although there are studies showing that the deficiency of DAW1L in stem cells can cause aneuploidy or like cell cycle arrest, the cells are still viable and they can remain pluripotent. So next we ask, is DAW1L important in cell differentiation? If you knock out DAW1L in mouse embryos, they will die before birth. And like the ones who they can't survive embryonic day 10.5, those who survived 9.5 will have severe cardiac effects. Like seeing in the mouse, it's like pale and no like blood vessel formed. And also in vitro differentiation assay, here this paper used embryo body differentiation, where it's undirected and the stem cells tend to differentiate into all three germ layers. And in this assay, you can see with the knockdown of DAW1L, you see the embryo body differentiation is compromised. So from the three different model, you can see that pluripotent stem cells can tolerate very low level of Cas9 methylation, but it loses the differentiation ability into somatic cells. And if the DAW1L inhibition can promote pluripotency acquisition, which is measured by reprogramming efficiency. So how does this phenotype is regulated? What is the mechanism of this regulation? So first thing we will ask is, does it affect the transcriptome? Does it have changes in gene expression? So in 3 different models, people have found that DAW1L deficiency will cause very little changes in transcription in the steady state transcriptome measured by RNA-seq. So first, in reprogramming cells, Coral performed RNA-seq on reprogramming cells with or without DAW1 inhibition. Despite we see a drastic change in the phenotype, we don't see many genes that are differentially expressed. Only like 300 genes are differentially expressed upon DAW1 inhibition. And similarly, in pluripotent stem cells and differentiating cells, very little like hundreds of genes are differentially expressed. And similarly in the RA differentiation. So the DAW1L deficiency don't cause big changes in don't cause big changes in steady state transcriptome. So next, if it's not through the mRNA, how does Cas9 methylation regulate cell identity? So from the literatures, we know that DAW1L mediated Cas9 methylation can be stimulated by other modifications like H2B monoubiquitination or H4K16 acylation. And it is also known to, there are evidence showing that it can inhibit Cas27 trimethylation in leukemia cells. So it has crosstalk with other modifications. So this led us to think that can K39 methylation function through interactions with other modifications? Like Rupa said, like does it function to make the cells, the somatic cells, more like stem cells, so that they can potentiate the cell to like prepare the cell to become pluripotent. And the first step to study how K39 methylation can regulate cell identity, we want to know where are they located at different regions on different cell types. So Coral performed ChIP-seq experiment for ChIP-seq for K39 dimethylation in somatic cells and stem cells. And we can see that most K39 peaks are found on the shared genes between MAPS and stem cells. And in those shared peaks, in those shared genes, you can see MAPS have a higher enrichment of K39 dimethylation compared to the stem cells. So they're in stem cells, they're kind of less on the same locations. So in order to investigate if K39 methylation can globally affect other histone modifications, Coral performed mass spec experiments on reprogramming cells with or without DOL1 inhibition. And from this heat map, you can see that if you compare the Day6 DOL1L inhibition group with the control, you can see most acylation levels are increased upon DOL1 inhibition, and which is very exciting to us because as we may remember from the earlier mass spec, the histone acylation is much more enriched in stem cell, and which is the key feature of stem cell epigenome. So we're thinking, oh, maybe the DOL1L can act to elevate the acylation levels in reprogramming cells, and that make them more like stem cells, so that facilitate reprogramming. And Coral also then performed ChIP-seq experiment for H3K9 acetylation, which is the most differentially enriched acylation in the heat map. And she found that the K9 acetyl enriched genes in the somatic cells, stem cells, and reprogramming cells with or without DOL1L are also like mostly overlapping. Like 7,000 genes will share, those shared genes will all have K9 acetyl enrichment. And if we take a closer look at those overlapping peaks, you can see that the DOL1L inhibition, which is shown in pink here, is higher than the control. It has higher enrichment of K9 acetyl near the transcriptional start site. So it does increase in the shared genes. And what's more, when we overlap the K9 acetyl enriched genes with the K39 dimethylation enriched genes, they're also mostly overlapping. So overall, this is suggesting that K39 methylation can inhibit K9 acetyl, and this is likely to be a local interaction. They're happening on like a large group of shared genes. So now I've shown data supporting that K39 methylation might inhibit active histone modifications and to make them more like stem cells. And what about repressive histone marks like K9 dimethylation that is also more enriched in somatic cells? So first, K9 dimethylation is a repressive histone mark that is found on the large domains. There are diamine-associated domains at the nucleus periphery, and it comprises of heterochromatin regions. A former graduate student, Zafira, analyzed some ChIP-seq data in the stem cells for K9 dimethylation, and she caught only 330 of those K9 didomains that are like very large tens of megabases, which is shown in the blue here, in the blue bricks here. And then she overlapped those K9 didomains with different histone modifications. we found it's not surprising that it's almost all of the canine dye domains will contain canine trimethylation as well, which is shown in the light blue, in the pink ticks here. And it is also, we also found like most of the canine dye domains can also contain active histone modifications K4 trimethylation here. So it's not excluded from this domain. And it is the K36 trimethylation, which is also found on active gene, on the gene body of the active genes. It also have overlap with canine dye domains. So what is exciting to us is that we found K79 dimethylation. It's only found in a very small proportion of the canine dye domains. They're like almost found in between the canine dye domains. And so we're thinking K79 dimethylation is especially excluded from those large domains. Does it have anti-correlation? Is it acting like, is it repelling the canine dye methylation? So this background this background led us to my central hypothesis, where K79 methylation can function through crosstalk with both active and repressive histone modifications to regulate cell identity. And unfortunately, current study in early development has been using the DAW1L loss of function, like inhibition, knockout, knockdown approaches to investigate this function. And to fill this gap, I want to learn more about the function and study the direct interactions. I applied the novel gain of function approach to investigate the function. So first, I developed stem cell lines that can overexpress a flag tag DAW1L in either wild type or with the catalytic dead mutation. It's like this function one, and they can be induced by a DOX-inducible promoter, and they integrated into the stem cell in a single site. And with the cell line, first in the Western, we can see that I first confirmed the expression of my tagged protein. And upon the DAW1L induction, there's an increasing amount of K79 methylation in all levels. And whereas in the catalytic dead mutation, there's not that increase. So I created a cell line, and I would use the cell line to ask, does global K79 methylation increase or affect polyphenol maintenance and differentiation? So if low level of K79 methylation is required in stem cells, and the cells have to get rid of them for polyphenol acquisition. So is it true that if we increase the K79 methylation, that will induce the polyphenol stem cells to trigger their differentiation and let them induce them to exit polyphenol state? So I will use self-renew assay, AP staining assay, and RT-PCR for polyphenol marker. And I want to see if the increase of K79 methylation will cause the cells to exit polypotent state. They are no longer pluripotents upon the DAW1L overexpression. Then, if it, regardless of the pluripotent maintenance, is it facilitating the cells to differentiate? Does having more K79 methylation will help the cell differentiate, even faster or have higher efficiency? And I'll use cell number or germ-layer marker gene expression to see does it have a phenotype in cell differentiation? And I would expect if the overexpression can help, this should have faster or higher differentiation efficiency. In parallel, I also use RNA-seq to find any changes in transcriptome upon the DAW1L overexpression in the pluripotent cells and differentiating cells. And in combination with information of the ChIP-seq for K79 methylation, I want to see is there a correlation there? Is the increasing amount of K79 methylation causing transcriptome at the certain locations, or is it an indirect effect? And from our preliminary data, K79 methylation could potentially interact with other modifications. It's shown some evidence about it inhibiting, it might reduce K9 acetyl, and it has an anti-correlation with K9 dimethylation. So we can ask, again, on those cells, I can do quantitative mass spec to see does it to detect changes upon DOMA inhibition. Is there other modification, other changes in the histone modification levels of other modification? And I'll also use ChIP-seq for K9 acetyl and K9 dimethylation to see are the changes, are K9 acetyl and K9 di changed at the location of K79 methylation, and to see if this is like direct effect. And so that was for global K79 methylation increase, which is not directed. It might happen at the location that we didn't want, or like we didn't expect. So to precisely increase K79 methylation at specific locations, I also built a cell model with dead Cas9 fused with the DOMA-L catalytic domain. And I used the Y type and again the catalytic dead mutation and integrate into the stem cell. And then I used the vectors containing guidanase targeting certain genes, or use the vector that's not targeting as a control, and induce and perform ChIP-seq experiments after harvesting. So here I use TWIS2 as a target from this enrichment, K79 di enrichment plot. We can see that TWIS2 is very lowly enriched in stem cells, but it can be enriched on the somatic cells. So I use this target and perform ChIP-seq using primers against the YWH-AE. This is the gene. This is the gene where it have high levels of K79 methylation in stem cells, and it's not targeted. But and also TNAG, which is a negative control that don't have K79 methylation enrichment in stem cells. So we can see that with the targeting, the white is showing the ones without guidanate targeting, and the gray is the one with the targeting group. So on the non-targeting locations, there's no difference between the targeting group and the non-targeting group, and there's a drastic increase of K79 methylation enrichment at the targeted location in the Y-type cell line. And it's more to show in the catalytic dead mutated cell line, still similar positive control and negative control, but this time we don't see an increase of K79 methylation on the target locations. So this is showing that we have a model that will like specifically increase the K79 methylation on our desired location. So using this tool, I could locally manipulate the K79 methylation level and detect changes of K79 methylation using like PCR. And here I can answer the I want to answer the question: does local K79 methylation increase can affect the K9 acetyl enrichment? It's like a direct manipulation. So I chose 5 genes that have K79 methylation and K9 acetylation, and I clone those guidanase into a plasmid with fluorescent and package them in lentivirus and then infect stem cells. Through sorting, I got the cells that are expressing guidanase and my transgene. And for those and from those cells, I can do a ChIP PCR for H3K79 methylation and H3K9 acetylation. So you can see if there is an increase in the K79 methylation level, and does that decrease the K9 acetylation? And also RT-PCR can help me understand, does that change affect the gene expression at the location? Similarly, I can target 5 genes that are found in the heterochromatin the canine dye domains. And I use the same targeting strategy, and I want to see if I can see the increase of K79 methylation in the canine dye domain. And does that deposition disrupt the canine dye domain, like does prevent the heterochromatin to be deposited at that location. So in terms of rigor and reproducibility, so I first try to design my experiment with proper controls, and I use catalytic inactivated DAW1L cell lines as my catalyst control. So the DAW1L is also known to form complexes and interact with other proteins. So using this control, I can determine, is it the result of K79 methylation or is the protein function? Also in chip PCR experiments, I will use controls to evaluate my chip efficiency with positive control and negative control. And also choose multiple gene locations for targeting in each experiment, and also use fluorescent protein to control for the transduction efficiency so that I can compare between my experiments. With that, I would thank everyone in the Sheridan lab, and Rufa has been a great mentor, and a postdoc, Coral, whom I based most of my research on the some experiments from Coral. And with that, I can take questions. So you're saying that the embryonic stem cells have very different cell cycle than induced pluripotent stem cells? No, I think. Oh, from differentiation. Oh. Oh. Yeah. Yeah. So from our RNA-seq experiment, so what Coral did was with chemical inhibition of DAW1L. From the 300 genes, I didn't take a closer look at those genes and see if there's cycling genes. No cycling genes.
Do you remember Coral's cycling data? Do you remember, has she done just cell count, and has she done exactly the facts for T1, T2 things? Do you remember data or not?
I think there's no difference, like upon DAW1 inhibition, there's no difference in cell cycle. I don't know about gene expression level. And I also, like personally, I performed experiments of the cell cycle upon my gain of function experiment. I don't see a big difference in cell cycle. But yeah, that could be something I can look for in my RNA-seq experiment. Yeah.
Yeah.
Yeah, we use FACS.
Yeah, by FACS.
Yeah, we use FACS to the. Yeah.
So it's not, it doesn't change that both the reprogramming was done by Coral and the overexpression was done by Xiaoya.
Yeah, I didn't see that difference. Yeah.
Actually, until we on the PNMSW by mass spec. It has been one of the most frustrating projects because it found almost 8,000 highly transparent genes modification, and there is no effect on steady-state transcription. There is no effect on either cell number, cell proliferation, the excluding by counting, or of the lengths like you're suggesting. There is no non-histone target of Doppler. So we also consider mass spec for anything that changes. See if it, that's exactly what we did. So we did with Lloyd Smith. We added the Doppler inhibitor, and then we looked for any protein levels that changed, and there were none to find the gap. And then we also looked, because Doppler actually stands for sorry, because I was like, what else is that? It's called disruptor of telomere function. It's actually yeast protein. Originally described in yeast, where it does something else. And so there's some related to telomere. So the alt pathway receptor, what is that? Everything was like, oh no. And then finally we did this mass spec, and there needs to be an ethical implication. One thing Xiaoya did mention, I think is important, is that none of the histone acetyl transferases or the histone deacetylases that perform all of those acetylations change in expression. So this is truly an epigenetic phenomenon that we get over.
Fantastic. This is dialogue. But in the interest of hearing a second really explains, I think you're going to have to move ahead. I'm going to ask Francisco.
Okay, so I'll be quick, so give Trevor time. And it's a pleasure to introduce Trevor Chamberlain. Trevor graduated from Utah Valley University. His advisor was Eric Domyan, who's an alumni from Genetics at JTC. He was a PhD in Samsung. And then he here in Utah, and then they worked in morphology and when he came to life, he was interested in a project that we just started at the time. Our laboratory study for development using the zero pressure model system. We also were intrigued about developing for the last couple of decades that used interspecies approaches to reprogram. Realized that there wasn't really a good system to analyze them systematically, closely phylogenetic space, so that they could be more understood. For example, you can take a glucose mouse egg and it looks like a mouse for a while, and then it figures out what does that mean. So we wanted to use essentially closely related fish species to try to understand those problems. And this has practical applications for reprogramming, that includes graft and regenerative medicine, or we're also interested in conservation genetics. It also has fundamental insights, for example, speciation, post-zygotic speciation have to do with the same boundaries we're looking at. And also more details on morphological change if you go more closely related in species. So Trevor has been tackling this. He's done an incredible job looking at bioinformatics. He's been pioneering bioinformatics in our lab. All the pipeline is all his. So I'm really, really impressed with everything he's done. As you see, it's gone very far from sequential construction. Also pioneering mitochondrial energy analysis and also cell lines. As I know, there's really very little work on cell lines, and he's developing those two. And so I'm going to leave the stage to him, but just want to say thank you for
All right, thank you, Francisco. So I'm going to go ahead and jump right into this. So I'm going to be talking about mitochondria today, and basically everyone here is probably familiar with mitochondria as their role in the powerhouse of the cell, and that's through their function in oxidative phosphorylation to make the energy currency of the cell ATP. But they're also involved in a lot of other functions listed here as heme synthesis, intracellular calcium signaling, and many metabolic pathways. And so they're a vital component to study for just about any cell biology. There's also some impacts in health that I'm not really going to go into. I'm going to skip right ahead to the parts that I'm really interested in my research are adaptation and speciation. And over here on the left, I am showing a study that was done that looked at these mammoths and samples collected across the globe. And what they did was they looked at the mitotypes of each of these samples and then looked at whether they were adapting to different environments based on where they were found and the timeline they were found in. And so the mitochondria, they do adapt to different environments that they're in, and you can see that in the genomes. And over here on the right, I have an example of a study looking at post-zygotic isolation. And post-zygotic isolation is when you have two different species that can still mate and produce an offspring, but there are boundaries to whether that zygote can survive or can be competitive. And what this is there's two populations of chameleons in Israel that have been separated by an ancient waterway. And these populations are distinct in their mitochondrial genomes. Their mitotypes are different. And so this study was looking at whether or not they can still look at mitochondria, maintain mitochondrial function if they do mate, and predicting that this is moving towards a speciation event.
Trevor, can you share slides?
And the reason why the mitochondria would be different is because they do have to interact with the nucleus. So in these hybrids that are inviable, what happens is you have the mitochondria that's all maternally inherited that can't interact with the nucleus or nuclear components in that hybrid and can't sufficiently drive that activity. And what I show here is the mitochondrial genome on the left. This is typical of most vertebrates. There's 37 genes, and 13 of those genes are protein coding. And I have those over here on the right put into these protein models in the oxidative phosphorylation complexes. And all the colored ones here are the mitochondrially encoded ones, and everything gray is coded for by the nucleus. And so you can see that these have to work together to be able to drive oxidative phosphorylation. And over time, you do end up with some incompatibilities that arise, and that's explained by this Dobzhansky-Mueller incompatibility model. And what you do is you start with an ancestral co-adapted population of the nuclear mitochondrial DNA. And then if something happens to separate that population, usually what happens first is you have divergence of mitochondrial DNA, and then you start having selection that acts on the standing variation in that population that's co-adapted to the mitochondria. And then you have further selection that acts on new mutations. And over phylogenetic time, you start having more and more incompatibilities that arise between those species or between those populations. And we're interested in this as a lab, as Francisco mentioned. We've branched out into this area of conservation. And what we're proposing essentially is biobanking biological samples of endangered species, and doing that by based on phylogenetics. And so this represents a gene tree of a bunch of species that you would want to biobank. And if you were to try and use these in a system for cloning, using one as a surrogate species and the other one as a donor, you could see that some of these would be competent. But somewhere along the way, you would have a phylogenetic barrier that's too great of a distance of relatedness, and you could no longer end up with a viable embryo. And so we're looking at whether or not we can find these compatibility boundaries. And the whole project is aimed at a mechanism of biobanking, where you biobank things in clusters, where you have, in green here, we have a non-endangered root species that would be biobanked together with endangered species, so that you, and do it close enough together so that everything is efficiently biobanked, to be able to work with that in a conservation setting. And the system that we're using is these fish that we have. They're Danionan fish is the name of them. We have these 12 species. We actually have a few more now that are in the lab. And what I show here is all the species that are pictured. I've sequenced, annotated those and assembled, I've assembled and annotated them. And then using a maximum likelihood approach in Raxamel and IQ tree, I assembled this tree. And on this tree, we have zebrafish at the bottom, and going up is going further related, or further distantly related from zebrafish. And then I have here colored, we have 3 genera. We have the Danio genus, the Divario genus, and the Microdivario genus. And what we're doing with these fish is we take species that, well, let's see, we take the zebrafish and we express embryos or eggs from that zebrafish, and we treat them with UV or gamma rays, and that cross-links the nucleus in that egg and effectively enucleates it. And then we take this enucleated egg and we fertilize it with sperm from one of the other species. And that creates androgenetic nucleocytoplasmic hybrid, or a hybrid. And these are haploid, but we use them as a surrogate for somatic cell nuclear transfer, like you would use in these somatic cell nuclear transfer experiments for cloning. And you can see these hybrids, like I said, were haploid. And this is demonstrating the haploid syndrome that you see in zebrafish. If you make a haploid zebrafish, you get this characteristic phenotype, where you get a reduced head, the yolk extension is reduced, the tail is reduced. They live for about 2 days, and you can study the development in that time. And this haploid syndrome is well characterized in the literature, and this is what we use for our controls. And you can see how that compares to a diploid zebrafish next to it. And then the hybrids that we've made in the lab, using the approach I described earlier, shows Danio alboliniatus looks very similar to the haploids that we make in zebrafish. But as we get more distantly related, Danio margaritatus and Divario equipinatus, or the giant Danio, they develop these defects. In this case, both of them are showing a yolk closure defect in which the tail, what will eventually become the tail, failed to close around the yolk, and that's a lethal mutation. Putting this into a phylogenetic context, zebrafish down here again, Danio alboliniatus is probably about 7 million years diverged from zebrafish, and then Danio margaritatus is about probably 11 million years, and with Divario equipinatus being about 20 million years diverged. So you can imagine that maybe there's one of the compatibility boundaries I described earlier, somewhere around the region between Danio margaritatus and Danio alboliniatus. Like I said, after I got all of these fish, the first thing I did was sequence them to try and explore whether or not there are incompatibilities. And I started with extracting genomic DNA. I amplified the mitochondrial genome through PCR, pooled those as amplicons, and then submitted that right here to the sequencing center and sequenced them with NovaSeq. And then the first thing I did, and this is going to be really hard to see on this screen, I apologize, but this is a plot showing all of the translated proteins of all 12 species at once. And this is on a circular plot, and the outside gray ring represents the zebrafish, Danio, and then going inward towards the middle is getting further diverged from zebrafish. And so a couple of patterns arise when I align things this way. Again, I apologize, it's so hard to see on here, but cytochrome B here shows what you would kind of expect with it getting more and more diverged, with more variance accumulating as you go towards the middle. But then you can see some other patterns in here, like ND6 has a big block right through here that just appears right away and doesn't gradually accumulate. And I would expect maybe that area doesn't have a whole lot of functional significance and can just have a lot of variation. Some other interesting things to note is the cytochrome C oxidase genes down here. Between all Danio species, it's very highly conserved. In fact, COX-1 has no variance between the other species, but there's a lot of single amino acid variants as soon as you make that genus jump into Divario genus. And cytochrome C oxidase 2 and 3 all show the same things there. And then finally, if we go and we look at the percent of variance by complex, you can see that complex 1 in almost every case has the most variance, but there's a couple of patterns that pop out there too, where again, cytochrome C oxidase in the Divario and microDivario species does have more variance than the other species. But complex 1 in Danio albulineatus shows a lot of variance. And I'll zoom in on that really quick. And this is ND5, which is a complex 1 gene in Danio albulineatus. And if you can see, there's a lot of variance, which are these magenta lines, that have accumulated in that species. When I first sequenced this, I thought this was a mistake. I thought I must have done something wrong, either in my sequencing approach or in my annotation. And so I wanted to talk a little bit about rigor and reproducibility when it comes to annotation and how to sort out these kinds of problems with sequencing. The first thing you do is you look at it and say, wow, is this a mistake? And so I did annotation originally just by hand, manually, because mitochondrial genomes are pretty small. And I just found the features by start codons, tRNA sites. I did some alignments and I did this. And then to double check my work, I started using this software, MitoFish and Mitos. And you can just take a mitochondrial genome, plug it right in, and it just kicks the genes right back out. And a piece of mind for that albulineatus gene was this matched my prediction of where that gene was with start codons and everything. And then to further investigate this, I did Sanger sequencing to make sure it wasn't just the samples I submitted and the amplicons, maybe they had some PCR mistakes in them when I submitted them to Illumina. So I do Sanger in smaller pieces around the genes. And I do this in everything that I kind of suspect. But then one of the things that I've discovered lately is the sequence read archive on NCBI. It can be a really powerful tool, especially when working with non-model organisms, because there's not a lot of genomes out there for these non-model organisms. The sequence read archive now has a lot of raw reads from a lot of the species I work with. And I was able to download the Danio albulineatus reads and then assemble the mitochondrial genome using a program called MitoBIM. Even sequencing that was done by another group and then assembled this way.
I just saw the email. Oh, forgot to.
Okay, well, if anyone's still in line, apologies, I guess this wasn't shared, so. Anyway, so for the sequence read archive, I was able to assemble the mitochondrial genome, and completely independent of the stuff that I worked on, I got the exact same genome as well. And I've done this to try and make sure that I have everything as accurate as possible. So, oh, this one's even harder to see in this lighting. The next thing I did was try and look, if I have all of these variants, do any of them have a functional impact on these proteins? And so the first thing I looked at was positive selection. And basically what I'm looking at is if I compare the non-synonymous to synonymous mutations, do they occur in a frequency that is greater than if I assumed it was under neutral selection? And what this means is there's an adaptive fitness for the places that are positively selected, and likely that plays a functional role. And so I used 3 different methods to look at positively selected sites: CODML, mean, and TreeSap. And I found 27 total sites that are under positive selection, and they're shown in pink over here. And many of them are shared among like all Danios, or I have one down here in ND4 that's shared among all Divarios, but some of them are distinct to individual species. And again, I wanted to point out this COX2, where I showed earlier, has a large number of variants. This also had positive selection in COX2. And the sites in giant Danio that have positive selection, I've shown here in this structure in yellow. And this part of COX2 is a binding domain. And what this binds to is cytochrome C. And cytochrome C has this heme center. And what that heme center does is it carries electrons in the electron transport chain. And when it binds to COX2, it transports that electron to the copper center that's in the middle of COX2. And so if I have positive selection here that affects the function of this site, it could impact the ability to bind to the nuclear encoded cytochrome C of another species. Looking even more at the cytochrome C oxidase complex itself, I did a covariation analysis. And this is something I did very recently on everything in this complex. And what I did was I got the nuclear genes that aren't part of my sequencing, but again, I used that sequencing read archive and a program called Hide Piper to build all of these genes that are part of this complex. And they're all nuclear encoded. And then I did the covariation analysis to see if I have a single amino acid variant in a mitochondrially encoded gene, does it also covary with a nuclear encoded gene that also has a single amino acid variant? And so that's what I'm showing here, is these are co-varying single amino acid variants in the giant Danio cytochrome C oxidase complex. This is all of complex 4. And so this looks like there's a lot of places that incompatibilities could arise if all of these function together. And I keep highlighting cytochrome C oxidase because when we hybridize the giant Danio to zebrafish, we get this phenotype. And this is compared to a wild type zebrafish that I show here on the left. And they all have this same, there's a reduced head region, there's a reduced yolk extension, reduced tail, and then you usually get this edema where the heart just expands really big. And then it also fails in the heart looping morphogenesis mechanism. And when I went and I looked up cytochrome C oxidase defects in zebrafish in the literature, I found the same phenotype, which was really neat to me to be able to see that you can induce this in zebrafish and get this same phenotype. And even if you compare levels of severity of this, you get similar levels of severity between the zebrafish-induced cytochrome C oxidase deficiency. And so this is where I'm trying to investigate at the bench. This is a good starting point, I feel like, for looking for a clear incompatibility. And I've been working on investigative tools like Francisco mentioned. I've been working in tissue culture, and I've spent most of the last year figuring out how to isolate cells, how to work with primary cells, how to, I've even been collaborating, trying to figure out if we can transfect RNAs into these cells. And I'm currently working on some assays as well. I'm working on a mitochondrial membrane potential assay, and this is an image I took showing these cells that have taken up this dye that gets pulled into the mitochondria when they have a healthy membrane. And so you can use this to compare samples and look at fluorescence intensity, and healthy mitochondria should fluoresce more than unhealthy mitochondria. Additionally, I've been working with a lab across the street trying to develop these respiratory functional assays, both in embryos and in cells, to look at mitochondrial function. The assay is called Seahorse. It measures oxygen consumption rate and extracellular acidification rate. And so this is where my work currently is, and my future directions going forward will be the data that I get from these. And in summary, I just wanted to go through and remind you, I found these incompatibility boundaries that appear probably around 11 million years at Daniel Margaritatus based on our cybrid experiments. I currently have all 12 of these species sequenced, assembled, and annotated, trees built, and I've been running functional tests with positive selection and covariation. And I've been finding some good indications that we do have incompatibilities between these species and zebrafish. One that I'm really interested in is the cytochrome C oxidase 2, and in fact, all of COX-4, or complex 4, the COX complex, in giant danios. And then finally, going forward, I'm going to measure mitochondrial activity in cybrids and hybrids and cell lines, doing this with the things I'm currently developing. And I think I'm out of time. I had a little bit more I was going to talk about. I think this is a good place to end. I was going to talk about another project that I've worked on involving this black-footed ferret. Just to kind of talk about it briefly, black-footed ferrets were nearly extinct in the 70s. And there was recently a black-footed ferret that was cloned using interspecies somatic cell nuclear transfer from a cell line that was stored since 1987. The parents of this cloned ferret are a black-footed ferret from the cell donor and an egg donor from a domestic ferret. And so the mitochondria was all contributed by the domestic ferret, and the somatic cells were from a black-footed ferret. And so we wanted to look at and see, could there be problems with essentially a cybrid created this way, even though it's a big step in cloning and conservation. And you can see that they're very closely related. They're within 1 million years diverged, very closely related. And so I don't have a lot of time, so 31 variants exist between them in protein coding genes, and 9 of 22 mitochondrial tRNAs have SNPs or indels. And this was significant because in humans, while tRNAs make up only 10% of a mitochondrial genome, about 40% of identified mutants in human disease exist in tRNAs in mitochondria. And so this could be a possible site where there could be problems. And to summarize this really quickly, I just wanted to say that we found this, and we've been talking with some folks at Revive and Restore about this, and trying to say that some care should be taken to ensure the mitochondrial health in cloned animals through this mechanism. Looking even more at conservation, this is my very last slide. I just wanted to also highlight some work that some undergrads of mine did while they were locked out of labs during COVID. They looked at this pipeline and they compared all of these species to start identifying patterns, because we are really interested in this conservation approach and looking at many species and seeing if we can account for mitochondrial health in these biobanking proposals going forward. And with that, I'd like to thank the lab, especially Francisco. He's been a great mentor. I thank my thesis committee, a few individuals that have helped me with tissue culture, and also my funding sources. And with that, I'll take any questions. Yes.
So I have two questions. One is for those very far away species, the giant Daniel. How different are the ecosystems? Right? Does one live in brackish water where it needs mitochondrial to function really hard because there's less oxygen? You know,
this is exactly what I've spent a lot of time looking at this and reading about this is the giant Daniels. First of all, they're huge fish compared to zebrafish. They can grow up to about 4 inches. I've seen them at pet stores, just monster fish in comparison. And so that's a very big difference right away. But in the wild, they do also live in deeper water, faster running water, and more brackish water than zebrafish and the other species. To go along with that, the Daniel alboliniatus as well, that I showed that ND5 gene in, they tend to be all over in their range. Most of these fish kind of exist in just small areas within like India and surrounding areas around there. But Daniel alboliniatus is kind of just found north to south, just all over that part of the world. And I wonder if maybe they have adaptations to just fit into a lot of different places. So, yeah.
And my second question out of curiosity is just, have you looked at like Homo naledi or any of the other human species, you know, like the in-between ones, through your pipeline to see how different besides the Neanderthal, the newer genomes, I don't know if those mitochondrial genomes have been.
Oh, so like Homo. Yeah. I haven't looked at those. I've spent some time looking at primates because of some classic experiments done in the 90s between chimps and human cells in hybrid experiments, but I haven't looked at any of the hominid species at all. That would be really interesting, though. Yes. That's a really good question. Most of these proteins, if you look at the structure, they have all of those alpha helices just going up and down and up and down. Most of those are core structural components. So in my head, it seems to be that they would fit together structurally a little better. However, in proteins like ND2, ND4, and ND5, those have a lot of antiporters in them. And a lot of the variants that I've found in those proteins seem to fall along edges of the antiporter domains. And so I've wondered if maybe those would affect the function of the antiporters rather than the structural components. Yeah, that's a good question, because you can do that in cell lines a lot of times. I don't know if we could do that in an embryo or not, because it's hard to change like the exact energy input that it's getting. Whereas in a cell environment, you can give it exactly what it needs, right? No, that's a really interesting question. I don't know an answer for that one, unfortunately. Yeah, 4, 1, and 5. Yeah, 30. Yeah, 3 has some. Yeah, 4, 1, and 5 seem to see the most. Mm-hmm. No, they don't develop a swim bladder, which is what they need to be able to survive, and so they don't survive beyond like day 5. I'm not sure. I am interested in looking at whether or not they have proton leak, if that was, and I should be able to measure that using seahorse, which I'm working on getting cells to the right stage at the right time to be able to put through the seahorse assay. And then that's one of my first questions, is whether they have proton leak. Yeah. But does that kind of cover? Okay. Okay.
Let's thank both of our speakers again.